Welcome students. Program Bachelor of Science second year, subject Botany, semester 3, course code BOC 103, course title Plant Anatomy and Embryology. Unit 7, Embryo and Endosperm Seed Structure, module name Endospermous Seed. Myself, Ms. Pratiksha Dabholkar, Assistant Professor Botany in Ganpat Parsekar College of Education, Harmal, Goa. This module focuses on the classification of seed, endospermous dicot seed, and endospermous monocot seed. By the end of this module, the student will be able to describe the structure of dicot endospermous seed and describe the structure of monocot endospermous seed. My dear students, when we classify a seeds can be divided into two, dicotyledonous seeds and monocotyledonous seeds. Again, this dicotyledonous seed is divided into endospermous and non-endospermous. Similarly, the monocotyledonous seeds are again divided into endospermous and non-endospermous seed. We are going to focus only on the endospermous dicot and endospermous monocot seeds. Let us first understand the endospermous dicot seed. You can see the picture over here. It is an example of a castor seed. Now, a dicot seed, endospermous, is divided into three. The endospermous dicot seed is made up of three layers: seed coat, endosperm, and the embryo. We will learn these three structures in detail. The first is the seed coat. The seed coat is made up of several layers. The first is the testa. Testa is the outer hard black and white covering which you can see in the picture. The next is the caruncle. Caruncle is a knob-like structure. In this previous slide, you can see that these are the castor seed and the white color structure present over there is nothing but the caruncle. After caruncle, we have a hilum, which is a point of attachment of the seed to the stalk. Next is a micropyle region, which is a small opening present at the caruncle region. We have the raphe. Raphe is a ridge running along the length of the seed. The last layer of the seed coat is a tegmen, which is nothing but the inner membranous seed covering. So it will be present below the seed coat. The next region is the endosperm. It is a white fleshy mass. Once you remove the seed coat or the outer covering, you will get this region, that is the endosperm. Endosperms are nothing but the storehouse of cells. They reserve the food material over here. In this case, we are taking the example of castor. So the reserved food material is castor oil. The third region which makes the endospermous dicot seed is the embryo. Its endosperm surrounds the young embryo. An embryo consists of two cotyledons in case of dicots, and it has an embryonic axis. This axis is divisible into radicle and plumule. The radicle region gives rise to the root, whereas the plumule region forms the shoot region. Now we are going to learn the endospermous monocot seed. Just like dicot, this endospermous monocot seeds are again divided into three: seed coat, endosperm, and the embryo. Embryo can be again classified as a single cotyledonous embryo and has an embryonic axis. In case of dicot, it had two cotyledons. In case of monocot, it has only one cotyledon. Let us first understand the seed coat. It is also called as the grain coat. They are thin, single layered, and they are fused with the wall or the grain wall, which is the pericarp. Next is the endosperm. This is a starchy region. This is a starchy endosperm in case of this maize seed. This region lies above the epithelium cell, and the function of this endosperm is food storage. The third region is the embryo region. It lies at the lower side of the epithelium. It is differentiated into one single cotyledon, which is protected by a shield-shaped scutellum. You can see from this picture, this is a shield-shaped scutellum. Embryonic axis, the upper embryonic axis is divided into again the upper region, which is the plumule, and the lower region, which is the radicle. The plumule is protected by a protective sheath called as the coleoptile. You can see over here, it is the coleoptile is present, which is protecting the shoot apex, or which is the plumule. Radicle is a protective sheath which covers the coleorhiza. So you have the radicle, which has again one region, which is the root cap, and below this root cap is the coleorhiza. Let us just summarize this entire endospermous seed. We first learned about the endospermous dicot seed, which has the seed coat, endosperm, and the embryo. The seed coat has got 6 regions: testa, caruncle, hilum, micropyle, raphe, and tegmen. Endosperm is a fleshy mass which stores food material. Embryo consists of embryonic axis and 2 cotyledons in case of dicot. Monocot again has got a seed coat, endosperm, and an embryo. The seed coat is a thin structure which is fused to the grain. Endosperm is starchy endosperm, and the function of this endosperm is storage of food. The embryo is differentiated into a single cotyledon and an embryonic axis. The single cotyledon is protected by a scutellum. The shoot apex or the plumule is protected by a coleoptile. The radicle or root apex is protected by a root cap, and the entire radicle is protected by the coleorhiza. These are some of the references which you can refer for this module. Thank you.